Welcome to Atoms Not Bits, Hard Tech Weekly, and we're here with the Tetsuwan team. Could you introduce yourself and what are you building?
Sure. My name is Cristian Ponce. I'm the CEO and co-founder at Tetsuwan. I'm coming from Caltech Bioengineering, where I worked on full genome engineering research. And Theo and I are building AI scientists.
Yeah, I'm Theo Schäfer. I'm the CTO of Tetsuwan. My background is in robotics. I studied EPFL and ETH Zurich, autonomous systems. I've done some research at MIT and at NASA JPL. And very excited to be here today.
Amazing. And your approach focuses on translating scientific intent into robot execution. Could you talk more about that?
That's correct. So the nascent field we belong to of automated discovery or AI scientists is interested in automating the scientific method. And scientific method has some bare bones building blocks. There's hypothesis generation, coming up with an idea based off of our understanding of our world, testing that hypothesis through experiments, and then looking at the experimental data to refine our model of thinking and come up with new hypotheses. So we're at the point where we do experimentation. When you come up with a hypothesis and a set of experiments to test that hypothesis, then usually a human scientist translates that into execution and then looks at the data. We want to be able to translate experimental intent into execution so that robots, automated platforms, could take care of this step. Humans come up with the ideas, and then automated systems execute those ideas. And humans collect and gather the data so that they're able to refine their thinking and come up with new, more innovative ideas instead of being caught up in the manual labor of the process.
That's great. And you're breaking a lot of bottlenecks in the life science space. I want to know, what does the day in the life of a lab scientist look like before and after deploying your program?
Well, unfortunately, I could tell you because at the bench, I didn't enjoy the work. And it was a large inspiration for Theo and I to work on this problem. I think a lot of people who are stuck in academia like to think that it's different in industry, or we cope and say, after grad school, it's going to be different. In academia, at least, the day of a scientist in a bioengineering lab is mostly spent at the bench, unless you're on the dry lab side. You're taking care of cells. You're doing really repetitive tasks. If you're doing genetic engineering, you're most likely doing some sort of cloning work. So you're doing the same subset of experiments all the time, over and over. You're doing your Gibson assembly, you're doing your transformations, you're doing your colony picking, you're doing your library prep over and over and over. And there's little variations. Maybe sometimes you tweak the parameters a little bit, but you're pretty much going through the same motions all the time. It was my understanding that, in industry, we don't waste our brightest minds at the bench. That would be ridiculous. They're sitting there coming up with the novel therapeutics that are going to go cure cancer. But then we go out and we talk to a whole bunch of different labs. We spend time with a bunch of scientists at the bench. And what we see is the same waste of time that we see in academia. The first lab that we deployed to in San Diego, they're a rare disease gene therapeutics lab. They make these RNA therapeutics for people with rare diseases. And we got to spend a lot of time with their two PhDs that are down there in the wet lab. And unbelievably talented and bright people, but they're sitting there at Excel sheets all day and with an 8-channel pipette doing 384 well RNA extractions and qPCR plate preps, just going all day. It was unbelievable to us that our brightest minds are being wasted. That is the life of a bioengineer at the bench. Your talent is a lot of times wasted on these repetitive tasks.
Wow, that's really insightful in terms of being on the ground. In terms of the lab automation space, lab automators, there's not a lot of them in the space, right? So what was that customer discovery process like to understand, their day-to-day, those rare people that can translate scientific language into code?
It's a really great question. Our thinking on lab automation has largely been shaped by how lab automation has existed for the past 30 years. When we meet these people, it's always in the context of high throughput, low differentiation automation. That's what automation has been used for. So there are not a lot of these people, and they're very restricted in what they can do because their job is very, very hard. This process of converting scientific to intent to executable code, interfacing what scientists want to do into something that a liquid handler or another lab robot can run is so intense and expensive and takes so long that this is only really justified for doing something that's going to be done at an ultra high throughput and a low differentiation at like a factory level. So a lot of our customer discovery and talking to lab automation engineers was shaped by these kind of incentives that biotechs have to only utilize automation for the purposes of high throughput, low differentiation tasking. So we speak to lab automation engineers and they have a lot of frustrations because when you hear lab automation, when a scientist hears lab automation, they think of, a robot helping them with their work, but that's not what it is. It's high throughput tasking. It's like manufacturing. So lab automation engineers, they're in lab and scientists are asking, hey, can you help me automate this protocol? I'm only going to run 100 times. But these lab automation engineers are overworked. They already have a lot on their plate, and there's a lot of misunderstandings about lab automation that make their job really tough. So, like a lot of our customer discovery was shaped by the way that things are. But our hope is that if we're able to automate this process of converting scientific intent to executable code, then lab automation can be justified for higher differentiation tasking, and it unlocks a whole new set of use cases for automation.
Wow. In terms of the manufacturing like tasks you talk about, like what possibilities open up in terms of high variety experimentation? What becomes accessible?
I'll give you a super simple example. This was actually our first toy experiment where we realized that there was more potential than the space had demonstrated. Late last year when GPT-4 Turbo was presented at OpenAI's Dev Day, the first thing Theo and I did was go and throw in a picture of a gel electrophoresis run into the model and ask it things like, I just asked it, are there bands here? That's it. What lanes have bands? But it told us not only the lanes that have bands, but the size of the different fragments, because there was ladder there. It told us that it caught a primer dimer, and it had like multi-step like scientific reasoning. It said this is a primer dimer. It's due to non-specific amplification, and that's due to bad annealing temperatures. So this is all to say that like these advanced like scientific reasoning capabilities that models are gaining allows us to approach high differentiation tasking. So if you could only do one thing over and over the same exact way, that's great for high throughput screening. That's great for running the same diagnostic PCR. But if you were able to troubleshoot and react to data and do things differently, then that opens up completely new experimental workflows. Something as simple as a PCR where you're kind of iterating on the parameters to get good amplification, or maybe something even more complicated that involves multiple experiments chained together, like a cloning workflow. That all becomes possible when you have something that's able to react to data and make decisions and edit its own code.
And what are the primary challenges with developing an AI that understands experimental context? I imagine there's many, but could you talk about some?
Sure. Basically, one of the very interesting problems that we're facing is the peanut butter jelly sandwich problem, right? Understanding the scientific intent properly, and there are a lot of implicit steps in a protocol that a scientist wouldn't write down, and they would just seem like common sense to them. And this is the issue that any programming problem faces is, you need to use your system and dig further, right? You're gonna have to ask the scientist more and more questions like, are you sure you gave us all the steps for a protocol? You run it in simulation and you realize that one of the well is overflowing, and you ask the scientist, you get your LLM to ask scientists follow-up questions this seems like there's a mistake here, and scientists will say, I forgot to tell you, you need to vacuum your filter so that it empties the well. That's a very good example, trying to understand and have this system. And this was not possible before GPT-4 and those large language models that enables us to simplify this scientific intent to executable code problem.
We can give you a real horror story on this too. This implicit type of stuff that a scientist has in their head, but it's tough for them to translate. Even when we have written protocols, like Theo was saying, there are so many implicit steps, right? And we like to think that of course we're good at documentation. Why would we be bad? That's the foundation of science. Good documentation, reproducibility, right? But all these implicit steps pop up. At our first deployment, there was a specific buffer that crystallizes. And the pipettes were left in it too long, so the buffer crystallized around the pipette, and then you pull the pipette back up, and then the pipette's blocked because there's just a whole bunch of crystals around the tip. Like, we could have never have known this, and the scientists didn't write in their protocol that make sure you don't leave that too long in the Gitsi wash buffer. So it's like all these little things, like Theo was saying, that we have to pull out of the scientists that they don't think a second time about.
Yep. There's so much nuance and so much context that's just stuck in their head that might not be written down on a piece of paper. Yep. That's amazing. So could you go back to San Diego? You know, you're in the biotech hub of the world doing testing, right? San Diego, the place where Illumina was built in its garage, you know, down there with some of the top scientists in the world. Could you talk about your first deployment there and the capabilities and your experience thus far?
Yeah, no, we're happy to. So we really wanted to target the highest volume of bench work that they were doing and the experiments that were involved in that. So this rare disease lab, they make a type of therapeutic. We're investigating a type of therapeutic called an antisense oligonucleotide, which is just a really, really overly fancy word for a oligo. So like a sequence that's of RNA that's only about 20 to 40 nucleotides long, that's able to go into the cell, find mutated pre-mRNA, bind to it or digest it, and stop it from becoming some bad protein. So it's really, really cool. It's like a little genetic missile. Goes into the cell, finds the target sequence, and then boom, like destroys it so it can't become something bad. The issue is that these ASOs, as are a lot of RNA therapeutics, are really, really tough to deliver. So there is, they call it the delivery problem of finding the right conjugate, which is the molecule that binds to the ASO and acts as the key to get into the cell. It binds to a receptor, and then the little missile goes in. But it's really, really tough to find these keys. To test out these ASO conjugate pairs, these scientists at La Jolla Labs, like you say, really bright people, like some of our top minds, are trying to see the impact that that has on the expression of certain genes. But the reality of doing that is doing a whole bunch of RNA extractions and doing a whole bunch of qPCRs over and over and over again to look and sift through all of these possible combinations and see what works and see what doesn't work. So what we built for them was a way to completely automate this process, to extract the RNA from the cells that they're dosing, and then prepare and design the plates that they're going to use for their qPCR, so that they can go from a treated cell, a plate of treated cells, all the way to data that tells them the efficacy of their different ASO conjugate pairs and the experiments. So this abstracts away a lot of the bench work. And this comes back to the question you were asking earlier. How does what we're building change the reality for a bench scientist? A scientist should be focused on coming up with new ideas, looking at data. They shouldn't be concerned with doing labor at the bench. So this solution to La Jolla Labs enables scientists to think of maybe a potential new mechanism or a new idea to approach instead of doing this work at a bench. It augments their labor to focus more on the intellectually stimulating parts of their job instead of the physically repetitive ones.
Yup. And for our last question, I want to zoom out here, right? What does the job of a human scientist look like in 2040? Could you paint that picture?
We have our logo, and I guess we've never explained much why it is the way it is, but it's just two triangles that are stacked on top of each other like this.
Okay.
One on the bottom and one on the top. And we have this bottom one because we feel like that's what science is today. There's a few people on the top, whether they're PIs or senior staff scientists, that control hypothesis generation and what people do, right? It's a very coveted position to be able to sit in like an armchair and read literature and come up with ideas and then assign scientists under you to go do the constituent experiments to test your hypotheses, right? There's only a few people who have that privilege to do real science today. But the future that we imagine in 2040 is that every scientist, every human scientist plays that role of the PI. They're the ones reading literature, coming up with ideas, going to conferences, and they are sending out the constituent experiments to labor, just like scientists do today, PIs do. But that labor is not human, it's robotic. So every scientist becomes a PI, and their army of grad students or staff scientists or RAs are just robots. So that they get to focus on what's really important.
That's amazing. And I imagine many of these scientists, when they came into industry, that's what they really wanted to do, right? They really wanted to be like a PI.
And it's interesting, despite that being like the hope of a lot of people who are coming into bioengineering, there's a lot of like very strange like cultural things that we've found in the lab. Like one of the biggest barriers to adoption for us is like the culture that's in labs. Like sometimes older scientists feel like if I did my PhD and it lasted 6 years, and I spent 6 years on the bench, and then I came in and I was like the lowest level like staff scientist, and I spent another 5 years on the bench, everybody should have to do that. We've heard it compared to a residency. It was a miserable time in my life where I was just spending it at the bench. So every single young scientist should have to go through that. So despite that being like, the hopes and dreams and ambitions of young scientists getting into industry, like to do groundbreaking work and develop some new therapeutic, and do like high level intellectual work, like there are some interesting like cultural attitudes of I went through this, so you have to go through this, or even distrust, rightfully so, of completely novel systems.
Yep, I could see that kind of. You have to cut your chops, on the ground at first. But you're unlocking science pretty much for all scientists, which is an amazing approach. Well, thank you so much to the Tetsuwan team. Amazing company out of SF, already deployed. Thank you again.
Of course, pleasure to meet you, Nolan.
Thank you, Nolan.